All right, welcome back. This is Lab 6, Darkfield Microscopy. So in the last lab, we had fully assembled a Kohler microscope, but for brightfield illumination, where our contrast was dark compared to our background. We're going to be inverting that optically in this lab with darkfield microscopy. So the first thing that we have to consider with darkfield microscopy is there's going to be restrictions, as you should remember from your exam, on the numerical apertures of our condenser and objective lenses. Now, these are actually labeled condenser and objective, but today we're not going to be using them in those roles. We're actually going to be flipping them such that the lens labeled objective is in fact our condenser, and our lens labeled condenser is in fact our objective. So we're going to be figuring out why that is mathematically before we begin assembling anything. So what you're going to be asked to do is calculate the relative numerical apertures of both of these lenses. So in order to do that, we've provided you a nice diagram on your labs that sort of relates the focal length of a lens to its half angle and its diameter. And from that, you should be able to calculate numerical aperture. So you should know which of these lenses needs to have a greater numerical aperture for darkfield microscopy. Does our condenser lens need a greater numerical aperture, or does our objective lens need a greater numerical aperture for darkfield to work? So you should be able to answer that, and then we're going to calculate, okay, so what are the numerical apertures of these objectives, or of these lenses? So you should go through and derive that formula theoretically to calculate the NA just from knowing the diameter and the focal length of a lens. I will provide you with the diameters now. So for our lens that's labeled objective, our total clear aperture looks to be about 24 millimeters, right? That is the diameter of the light that it allows through, okay? Our lens labeled condenser, our diameter is 22.5 millimeters, right? So that is the diameter of the clear aperture where light is allowed to pass through. So you should already know these focal lengths from Lab 2. Go ahead and reference those if you don't have them handy. And using that formula that you derived, go ahead and calculate what is the numerical aperture of each of these lenses. And I'll go ahead and pause the video for that. So now that you've calculated the numerical apertures, you should know that obviously our objective lens, the one labeled objective, has a greater numerical aperture than our condenser lens. So we have to use this lens labeled objective as our condenser, because in darkfield microscopy, you need a higher angle of illumination that cannot be collected by your objective. So we actually have to sacrifice a bit of resolution by using our second best lens as our objective lens, which does, again, cut down on the total amount of light that we let through and the resolution of our total resolution of our system. So finally, for darkfield, you do need an annulus. This is going to be placed somewhere behind our condenser lens that we are actually using, the objective lens that's being used as our condenser in the system. We will find a place somewhere behind it that we will use to restrict out the lower angle light and let through just a annular sliver of light. So I will go ahead and measure these dimensions. So an annulus has an inner and an outer diameter. So the outer diameter is about 24.5 millimeters. So that is the distance from the outer edge to the outer edge. And the inner diameter is 23.5. So that is the length from the inner edge to the inner edge. Right, so this was specifically designed in mind to be used with this lens. So you go ahead and record those down, and now you sort of have all of the information you need to do a proper ray tracing diagram of this Darkfield microscope, even though I haven't assembled it yet. We're not going to be using our iris today because our condenser will effectively, our annulus will act as our iris because we only want a very small sliver of light outside getting through. And we have our nice little collimated light source. We're going to put our annulus, our condenser lens, our sample holder, our objective lens, our tube lens, and then finally our camera. So as I am assembling this microscope, go ahead and draw out using the graph paper on the lab that we provided for you, the ray tracing diagram for this Darkfield microscope, and so you can pause the video. When we come back, we will have this microscope set up, and I will go through sort of the more finer alignments. All right, so hopefully you have made a nice ray tracing diagram of what you expect this Darkfield microscope to look like. In the meantime, I've been building and aligning this. You can see we have the objective lens in the condenser position and the condenser lens in the objective position. Again, just to make sure that our condenser has a higher numerical aperture than our objective, and in our kits, this is the highest numerical aperture lens that we have. And I already have mounted a sample of my hair. You can see that it looks similar to what we've seen in the previous lab, where we have a bright background and dark foreground because the hair is effectively just absorbing the light. It's not going to look as crisp because our condenser has such a high numerical aperture that it does tend to under-illuminate in terms of the field of view, the total area that is picked up by our objective lens, but that's just a flaw in the system. What I haven't installed yet is this annulus, right? This is what is essential for giving us that dark field contrast. We're going to need to put this somewhere in between our collector lens and our condenser so that the image of this annulus as cast by the condenser is going to be perfectly out of focus on our sample, but still not collected by our objective lens. And you can see that obviously a lot of light is getting through our sample and it hits our objective lens, and you end up with dark contrast and a bright background as a lot of light is passing by this objective lens into our tube lens. So if we go ahead and install this annulus here, tighten that down, and I will try to position it. Right, so you should have probably determined that the optimal place for our annulus is in the back focal plane of this condenser right here. So you can see it's very sensitive to any sort of changes in alignment. I'm just trying to adjust it so that my background is as dark as possible. Let's try to move to, yeah, I seem to have lost my hair in that installation, but there is my hair once more. I will do a little bit of focusing just to make sure that that's nice and in focus, and you can now see that the hair is no longer dark on a bright background. You can see that the edges of the hair, especially that lower edge, is bright on a dark background. That is because the annulus is the image of this annulus is being cast by our objective lens onto our sample. And any light that doesn't interact with our sample just passes by and is not collected by our condenser lens. You can actually see that there is a sort of nice wide annulus of light that is missed, or that misses our objective lens here. So the light that's actually coming out of this objective, while it's very little, does contain the information of the diffracted light as it passes near the hair. So that light that gets really close to the hair will diffract, and some of it actually diffracts into this lens, and then is effectively just collected by our tube lens. And you'll notice that there's something that I haven't talked about yet, which is this gray tube. This is actually the namesake of the tube lens. So in Darkfield and in other modes of microscopy where you have positive contrast, which is to say that your sample is brighter than your background, you want to minimize any stray light hitting your pixels or your image plane as possible, because your sample here is very, very effectively dim compared to what it would normally be like in darkfield or in brightfield contrast. So any stray light that makes its way from any sort of light sources in the room, or even stray light that makes its way, past all of our lenses, it might, scatter off of the floor or the ceiling and then make its way into the camera lens. That might be enough to drown out our small amount of signal from our diffractive sample. So in order to prevent that, we have this tube which will effectively only let in light that's passing through this tube lens onto our image plane. So just a way to sort of maximize contrast in your dark field setup is to use this tube. Sometimes you will even see people using paper towel tubes that work really well, just as a sort of way to block out any stray light that might make its way past the tube lens. So while hair is a nice sample to sort of illustrate the absolute differences in bright and dark contrast, it's not the most biologically active or interesting sample to look at. So I have prepared, as with last time, another sample of pond water. And we will see the differences that we can pick up with as we illuminate with Darkfield. So let me slide this in there. And there's always interesting stuff to find in the pond, even if it's just plant life. Let me move this around and get something in focus. So you can see I've picked up a little bit of probably algae, and you can see that it is bright on a dark background. So, and you can actually see I have not probably perfectly aligned my my annulus, because there is a place I sort of inadvertently put my finger in this in the infinity space between our objective lens and the tube lens, and you can see that that actually makes a small difference, at least in terms of the contrast. Yeah, obviously this is I'm losing quite a bit of information by doing this, but I'm at least sort of increasing the contrast. So there's definitely a little bit more that I could probably do to align this annulus. And you can see it's very sensitive as I bring this forward and backward. You really increase the intensity of that background. Right, and if I remove this annulus and we effectively switch back to brightfield, you'll see the difference that it makes. We'll end up with very, very dark algae, because again, algae is really photosynthetic. It has lots of pigments in order to absorb all that sunlight that it needs. And it's quite dark in this brightfield setup against that bright background. So a lot of the color actually gets washed out in this case. But again, thinking back to your exam, Darkfield has one glaring fundamental flaw, which is that we have really put a upper limit on the numerical aperture of our detection optic. So our light gathering capacity and our resolution take a huge hit by restricting exactly what light we're able to, how much light we're able to collect on the detection side. So while Darkfield is quite sensitive, and you would be able to really pick out very small scattering samples with ease, you're not going to get that same resolution as if it's in the previous lab, where we had these two objectives switched, or we had our higher NA optic on the detection side, because that's really what brings you the crispness that's necessary to resolve finer structures, even though we're sort of able to detect them. With Darkfield, we're able to sort of see any sort of scattering that goes on. We're not able to really resolve the finer details as well. So that concludes our Darkfield lab. Next, we will be looking at another way to generate positive contrast, which is with a polarization microscope, where we'll be introducing linear polarizers into our setup in order to look at how samples will rotate the polarization of light.